Welcome back, friends. Welcome back again to one more other lecture session from this channel. So in this lecture session we will be studying regarding meiosis and its regulation from cell cycle and cell division in detail. So here many of the students, they might feel some inconvenience in understanding this particular concept, but if you pay your attention, just it becomes a piece of cake for you people to understand. So here what is meiosis? Meiosis, it is a type of reductional phase or reductional division which is most commonly observed in case of reproductive cells or in case of sex cells. So here why the specific meiotic division, it is termed as reductional phase or reductional division, because the specific meiotic division, it is responsible for regulating the reduction in the chromosomal number in case of the daughter cell. So the reason why the specific meiotic division, or it is otherwise known as reductional phase or reductional division. So here mitosis, which is most commonly observed in case of somatic cells or in case of vegetative cells, mitosis, it is termed as equational division because the specific mitotic cell division, it is responsible for regulating the constant chromosomal number both in case of the parental cell and even in case of the daughter cell. But whereas meiosis, it is responsible for regulating the reduction in the chromosomal number in case of the daughter cell. So the reason why meiotic division, or it is otherwise known as reductional phase or reductional division, and especially the specific meiotic division, it is observed in case of reproductive cells or in case of sex cells. And here the term meiosis, for the very first time, it was coined by Farmer and Murray. Is it clear? So let me write it on the board. Meiosis is a type of reduction phase or division which is most commonly observed in reproductive cells or sex cells. And here the term meiosis, for the very first time, it was coined by Farmer and Murray. Is it clear? So let me write it here. The term meiosis for the very first time was coined by Farmer and Murray. So here the specific meiotic division, it is responsible for giving rise to an offspring. So here the parental organism, it is capable to give rise to an offspring by taking the advantage of a specific cell division termed as meiotic division. So here meiosis, it is responsible for ensuring the haploid phase in the life cycle of a living organism, but whereas fertilization, it is responsible for regulating the fusion of haploid gametes. Is it clear? Meiotic division, it is responsible for ensuring the haploid phase in the life cycle of a living organism, but whereas fertilization, it is responsible for regulating the fusion of haploid gametes. So here, in order to understand this particular concept, here we need to take the advantage of the basic concept here. So let me show it here. So as we all know, in case of humans, from the male father has chromosomal content S2N, right? Male father, right? Chromosomal content is 2N. Then in the same way, female mother has chromosomal content as 2N. So here, if the 2N from the father and the 2N from the mother, if they undergo fusion, it leads to the formation of tetraploidy condition. Never and ever in tetraploidy condition, it is capable to give rise to an young organism. Always the specific 2N condition, it is responsible for giving rise to an young organism, because all the cells in case of humans, they are diploid in nature, they are not tetraploid in nature. So here in order to regulate the production of and diploid cell, so here the specific chromosomal number, it has to undergo reduction. When the specific 2n condition, when it undergoes reduction exactly to half, 2n becomes n. Then in the same way, when the 2n from the female, when it undergoes reduction exactly to half, 2n becomes n. So here, once when both the haploid gametes, once when they undergo fusion, it leads to the formation of a 2n product termed as zygote. Zygote, right? So here, after the successful formation of zygote, now here the specific zygote, it undergoes repeated mitotic divisions in order to give rise to a young organism. So here meiosis is responsible for ensuring the haploid phase in the life cycle of a living organism. But whereas fertilization, it is responsible for the fusion of two haploid gametes in order to give rise to a 2n product termed as zygote. Is it clear? The specific mechanism or the specific process through which the chromosomal number, here it has reduced to half, here it is nothing but what? Meiosis. Is it clear? So let me write it here. Meiosis is responsible for ensuring the haploid phase in the life cycle of a living organism. But whereas fertilization is involved in the fusion of two haploid gametes. Two haploid gametes, right? So here meiosis or the specific meiotic division, it is responsible for giving rise to an young organism, or it is responsible for giving rise to an young offspring. So here, if we consider the specific meiotic division, it has certain salient features. So here, what are the various salient features of meiotic division here? Meiotic division, it has two rounds of nuclear division termed as Meiosis-1 and Meiosis-2 and a single cycle of DNA replication. Is it clear? So let me write it here. Some of the salient features of meiotic division may include meiosis involves two rounds of nuclear division. Meiosis-1 and Meiosis-2. Meiosis-1 and Meiosis-2. And a single cycle of DNA replication. And a single cycle of DNA replication. Right? So in general, the specific Meiosis-1 generally it begins or it commences after the successful organization of replication mechanism during the S phase of the cell cycle. Is it clear? So let me write it here. Meiosis-1 in general begins after the successful commencement of replication during S phase. During S phase. Is it clear? So here after the successful organization of the replication mechanism during the S phase of the interphase stage of the cell cycle, in general, the specific meiotic division, it comes into the picture. Is it clear? So let me arrange the things here. And the next specific salient feature of the meiotic division, here it is nothing but, in case of meiosis-1, genetic exchange, it occurs in between the non-sister chromatids of homologous chromosomes. So here, as I have already mentioned you, meiotic division, it contains two rounds of nuclear division termed as meiosis-1 and meiosis-2. And especially in case of meiosis-1, genetic exchange, it occurs in between the non-sister chromatids of homologous chromosomes. So here, as we all know, all the cells in case of humans, they are diploid in nature. So here, why all the cells in case of humans, they are diploid in nature? Because basically one specific diploid cell, it has chromosomal content as 2n. Is it clear? So let me show it here. One specific diploid cell. So let us assume it as a diploid cell. So here in case of the diploid cell, the chromosomal content here it is termed as 2n. So here why the chromosomal content here it is termed as 2n? Because one specific diploid cell, it has 23 pair of chromosomes or 46 chromosomes. So out of 46 chromosomes, 23 chromosomes are from the father and the other 23 chromosomes are from the mother. Is it clear? So let us assume it as the father chromosome, right? And here it is the mother chromosome. It is the mother chromosome and here it is the father chromosome. So here 23 chromosomes are from the mother and the other 23 chromosomes are from the father. And especially during the S phase of the cell cycle, when the replication mechanism occurs, under such condition, here the chromosomal number, it becomes double. That means initially the specific diploid cell has 46 chromosomes, but here during the S phase of the cell cycle, now here the chromosomal number in case of the diploid cell, here it is nothing but 92. Is it clear? Though the chromosomal number, it has become double. During the S phase of the cell cycle, but here we cannot consider them as individual chromosomes because both the chromosomes, they are in association with one single centromere. So the reason why here we cannot consider them as individual chromosomes, and here they are termed as sister chromatids. Is it clear? Both the chromosomes, they are in association with one single centromere. So the reason why here they are termed as sister chromatids. Especially in case of the meiosis-1, genetic exchange after the successful commencement of the replication mechanism during the S phase of the cell cycle. Now here the meiotic division, it comes into the picture, and especially in case of the meiosis-1, genetic exchange, it occurs in between the non-sister chromatids of homologous chromosomes. So here, what are homologous chromosomes? So let us assume it as the father chromosome, right? And here, let us assume it as the mother chromosome, right? Mother chromosome. So here these two chromosomes, they cannot be considered as homologous chromosomes because these two chromosomes, they are not similar in size, shape and even in case of the length of the DNA. So the reason why they cannot be considered as homologous chromosomes. Homologous chromosomes, they possess or they contain similarity in case of size, shape and even in case of the length of the DNA. So here the genetic exchange, it occurs in between the non-sister chromatids of homologous chromosomes. So let us assume one more other chromosome here. So here it is nothing but the mother chromosome, right? So here both these chromosomes, they can be considered as homologous chromosomes because both these chromosomes, they are having a similarity in case of size, shape and even in case of the length of the DNA. So here the reason why the specific chromosomes, they are termed as homologous chromosomes. So here these two chromatids of the concerned chromosome, they are termed as sister chromatids. Never and ever the genetic exchange, it occurs in between the sister chromatids of homologous chromosomes. So here the genetic exchange it occurs in between the non-sister chromatids of homologous chromosomes. That means in between this specific chromatid and this specific chromatid. So here these are termed as what? Non-sister chromatids. In between the non-sister chromatids of homologous chromosomes, genetic exchange it occurs during the meiosis-1. Is it clear? So let me write it here. In meiosis-1, genetic exchange occurs in between the non-sister chromatids of homologous chromosomes. Is it clear? And at the end of the meiosis 2, here we can observe 4 haploid cells. Is it clear? So let me write it here. At the end of meiosis 2, 4 haploid cells are formed. So here basically the specific meiotic division, it has 2 different phases. What are those? Meiosis 1 and meiosis 2. So here what exactly happens in case of meiosis 1? Meiosis 1, or it is otherwise known as heterotypic division or reduction division. So here why the specific meiotic division? Here it is termed as reduction division here, because the specific meiosis 1, it is responsible for regulating the reduction in the chromosomal number. So the reason why meiosis 1, or it is otherwise known as heterotypic division or reduction division. Is it clear? So let me write it here. Meiosis 1, it is a type of heterotypic division. Heterotypic division or reduction division. So here during the meiosis 1, here the separation of sister chromatid does not take place. In what way? In case of the mitotic division, the sister chromatids, they are capable to undergo separation because of the splitting of the centromere. Such specific separation of the sister chromatid, it is not observed in case of meiosis 1. In case of meiosis 1, separation of sister chromatid, it is not observed because in case of meiosis 1, splitting of the centromere does not occur. Is it clear? So let me show it here. So let us assume it as a chromosome here. So here both the sister chromatids, they are aligned by taking the advantage of one single centromere. So here during the anaphase stage, what exactly happens is splitting of centromere occurs. So here when the splitting of centromere takes place during the anaphase stage, under such condition, here the specific sister chromatids, they are capable to undergo separation. So here this is the scenario which is observed in case of the mitotic cell division. But here in case of meiosis 1, separation of sister chromatids does not take place because in case of meiosis 1, splitting of the centromere does not occur. And instead of the separation of the sister chromatid, in case of the meiosis 1, segregation of homologous chromosomes takes place. That means the specific genetic exchange which has took place in between the non-sister chromatids of homologous chromosomes, these homologous chromosomes, they are capable to undergo segregation or they are capable to undergo separation during the meiosis 1. Is it clear? So let me write it here. During the meiosis 1, separation of sister chromatids does not take place. and instead segregation of homologous chromosomes takes place. Segregation of homologous chromosomes takes place. Homologous chromosomes takes place. That means the genetic exchange which has took place in between the non-sister chromatids of homologous chromosomes. Now here, these two homologous chromosomes, they are capable to undergo separation during the meiosis 1. Is it clear? And then after, if we consider the meiosis 2, or it is otherwise known as equational division. So here, why the specific meiosis 2, or it is otherwise known as equational division? Because the specific meiosis 2, likewise the mitotic division, it is also responsible for regulating the constant chromosomal number both in case of the parental cell and even in case of the daughter cell. Is it clear? So let me arrange the things here. And as already mentioned, in case of meiosis 2 it is a type of equational division. So here, why the specific meiosis 2, it is a type of equational division here? Because in case of the meiosis 2, the chromosomal number, it remains constant. Instead of undergoing reduction, here the chromosomal number, it remains constant. So the reason why meiosis 2, likewise the mitotic division, here it is also a type of equational division. And here, in case of meiosis 2, separation of sister chromatid occurs. Because of the splitting of the centromere, separation of the sister chromatid occurs in case of the meiosis 2. In what way? In case of the mitotic cell division, separation of the sister chromatid occurs because of the splitting of the centromere. Even in case of the meiosis 2, because of the splitting of the centromere, separation of the sister chromatid occurs. Is it clear? So let me write it here. Meiosis 2, right? It is a type of equational division. It is a type of equational division. And here, in case of meiosis 2, especially the chromosomal number, it remains constant. In meiosis 2, the chromosomal number, it remains constant. And especially in case of meiosis 2, separation of sister chromatid occurs because of the splitting of the centromere. Here, in case of the meiosis 2, here we can observe the separation of the sister chromatid. Is it clear? Because of the splitting of centromere, separation of sister chromatids takes place. Is it clear? So at first we will be studying regarding meiosis 1. So here, what is meiosis 1? So as I have already mentioned you, meiosis 1, it is a type of heterotypic division or reduction division. And here, basically the specific meiosis 1, likewise the mitotic division, it has various phases here. So here, what are the various phases of meiosis 1 here? The various phases of the meiosis 1, it may include prophase 1, metaphase 1, anaphase 1 and telophase 1. Is it clear? So let me write it here. Meiosis 1, it has various phases which may include prophase 1, metaphase 1, anaphase 1 and telophase 1. Telophase 1. So here at first we will be studying regarding prophase 1. So here the prophase 1 of the meiosis 1, it is typically much more complex in comparison with the prophase of the mitotic cell division. So here in comparison with the prophase of the mitotic cell division, here the prophase 1 of the meiosis 1, it is typically much more complex. And here basically the prophase 1 of the meiosis 1, it has various stages here. So here what are the various stages of the prophase 1 of the meiosis 1 here? The various stages of the prophase 1 of the meiosis 1, it may include leptotin, zygotin, pachytin, diplotin and diakinesis. Is it clear? So let me write it here. Prophase 1. Prophase 1 of the meiosis 1 is typically much more complex than the prophase of mitosis. And here basically the prophase 1 of the meiosis 1, it has various stages like leptotin, zygotin, pachytin, diplotin and diakinesis. Is it clear? So let me write it here. Prophase 1 of meiosis 1 has 5 different stages, which may include leptotin, zygotin, pachytin, diplotin and diakinesis, right? So at first we will be studying regarding leptotin. So here what exactly happens during the leptotin? Is it clear? So let me arrange the things here. As I already mentioned, the prophase 1 of the meiosis 1, it is typically much more complex in comparison with the prophase of mitosis. And here, basically the prophase 1 of the meiosis 1, it has 5 different stages, which may include leptotin, zygotin, pachytin, diplotin and diakinesis. So at first we will be studying regarding leptotin. So here what exactly happens during the leptotin stage? So here during the leptotin stage, the compaction of the chromosome begins. That means the condensation of the chromatin material in general, it begins during the leptotin stage, which is a part of the prophase 1 of the meiosis 1. Is it clear? So let me write it here. Leptotin. So here during the leptotin stage, the compaction of the chromatin material or the compaction of the chromosome begins. So here the chromatin material starts remaining in condensed form. Right? So here because of the condensed form of the chromatin material during the leptotin stage, now here these specific chromosomes, they are clearly visible under light microscopic observations. Is it clear? Chromosomes are clearly visible under light microscopic observations. And here during the leptotin stage, which is a part of the prophase 1 of the meiosis 1, here the successful movement of the centrosome towards the opposite spindle pole takes place. Is it clear? During the leptotin stage, which is a part of the prophase 1 of the meiosis 1, successful movement of the centrosome towards the opposite spindle pole takes place. During the leptotin stage, movement of centrosome towards the opposite spindle pole takes place. Towards the opposite spindle pole takes place. Spindle pole take place. And here, especially in case of animal cells, here we can observe one of the unique feature which is termed as bouquet stage. So here in case of animal cells, all the chromosomes they aggregate together in accordance with the direction of the centriole. Is it clear? In accordance with the direction of the centriole, all the chromosomes in case of the animal cell they aggregate together. And here, because of the aggregation of all the chromosomes within the animal cell in accordance with the direction of the centriole, now here it resembles the shape of bouquet. So the reason why the specific stage here it is termed as bouquet stage. Is it clear? So let me show it here. Especially in case of animal cells. In case of animal cells, all the chromosomes. So here all these are nothing but what? The chromosomes, right? So here in accordance with the direction of the centriole, all the chromosomes they aggregate together, and here it resembles the shape of an bouquet. So here what is bouquet here? Bouquet is nothing but a bunch of flowers, right? Bunch of flowers or group of flowers, here it can be considered as a bouquet. In the same way, when many number of chromosomes, when they aggregate together in accordance with the direction of the centriole, now here it resembles the shape of bouquet. So the reason why this specific stage here it is termed as bouquet stage. So let me show it here. And here all these are nothing but the chromosomes. Is it clear? And here the specific stage is termed as bouquet stage. Bouquet stage, right? So let me write it here. In case of animal cells, during the leptotin stage, of the prophase 1 of the meiosis 1, all the chromosomes aggregate together in accordance with the direction of centriole. And here the specific stage is termed as bouquet stage. Right? So here this is regarding the leptotin stage, which is a part of the prophase 1 of the meiosis 1. And then after we will be studying regarding zygote stage in detail. Is it clear? So let me arrange the things here. So after the leptotin stage of the prophase 1 of the meiosis 1, the next specific stage of the prophase 1 of the meiosis 1, here it is nothing but zygote stage. So here what is zygote stage and what are the various salient features which are observed during the zygote stage? So here during the zygote stage, the two homologous chromosomes which are similar in size and shape, they start undergo pairing. And here the mechanism or the process through which homologous chromosomes they start undergo pairing, here it is termed as synapses. And here electron microscopic observations they have revealed that here in order to regulate the pairing, in between the two homologous chromosomes, here there is a requirement of a specific protein complex termed as synaptonemal protein complex. So here synaptonemal protein complex basically it is composed of 3 thick lines of DNA and many number of proteins. Is it clear? So let me write it here. Zygote in stage, right? So here during the zygote in stage, the two homologous chromosomes, which are similar in size, and shape, start undergo pairing. And here the mechanism or the process through which the two homologous chromosomes they start undergo pairing, here it is termed as synapsis. And here electron microscopic observations they have revealed that in order to regulate the synapses in between the two homologous chromosomes, here there is a requirement of a specific protein complex termed as synaptonemal protein complex. Is it clear? Synapsis. And here electron microscopic observations. Have revealed that synapses in between two homologous chromosomes, can be organized by the means of a specific protein complex termed as synaptonemal complex. Right? So here synaptonemal protein complex basically it is composed of 3 thick lines of DNA and many number of proteins. Is it clear? So let me write it here. Synaptonemal protein complex is composed of 3 thick lines of DNA and many number of proteins, right? 3 thick lines of DNA and many number of proteins. Is it clear? And here the first two stages of the prophase 1 of the meiosis-1, they are much more shorter in comparison with the next upcoming stage of the prophase 1, which is nothing but the packaging stage. Is it clear? So let me write it here. The first two stages of the prophase 1, are relatively shorter in comparison with the packaging stage. So here, because of the synapses or because of the pairing in between the two homologous chromosomes, now here the specific condition here it is termed as bivalent condition or tetrad condition. Is it clear? Because of the pairing in between the two homologous chromosomes. Now here the specific condition, here it is termed as bivalent condition. So here why it is termed as bivalent condition here? Because here we can observe two chromosomes. So the reason why here it is termed as bivalent condition, or in other terms, here we can consider it as tetrad condition. So here why it is termed as tetrad condition? Because two chromosomes, they has four sister chromatids. So the reason why the specific condition, here it is termed as bivalent condition or tetrad condition. Is it clear? So let me write it here. Because of the pairing in between two homologous chromosomes, a specific condition termed as bivalent or tetrad condition. Tetrad condition is observed. Is it clear? So here this is regarding the zygote stage, which is a part of the prophase 1 of the Meiosis-1. And then after we will be studying regarding the packet stage, which is a part of the prophase 1 of the Meiosis-1 in detail. Is it clear? So let me arrange the things here. So after the zygote stage, the next specific stage of the prophase 1 of the Meiosis-1, here it is nothing but packet stage. So in comparison with the leptotin stage and the zygote stage, packet stage, it is much more complex and even it is a significant stage. So here during the packet stage, crossing over takes place in between the non-sister chromatids of homologous chromosomes. So here the specific bivalent condition or the tetrad condition which has successfully formed during the zygote stage. Now here in between the non-sister chromatids of homologous chromosomes, genetic exchange or the crossing over takes place. So here the specific region where the crossing over takes place, here we can observe an X-shaped structure, and here the specific X-shaped structure, here it is termed as chiasmata. Is it clear? So let me write it here. In comparison with leptotin stage, leptotin and zygote stage, packet stage is much more complex and significant. So here during the packet stage, crossing over takes place in between the non-sister chromatids of homologous chromosomes. Crossing over takes place in between the non-sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. Right? Homologous chromosomes. So here the specific region where the crossing over has took place, here we can observe an X-shaped structure, and here the specific X-shaped structure, here it is termed as chiasmata. Is it clear? So let me write it here. The specific region where the crossing over has took place took place, an X-shaped structure is observed. X-shaped structure is observed, which is termed as chiasmata. Chiasmata, right? And in addition to this, even we can observe one more other specialized structure at the specific region where the crossing over has took place, and here it is termed as recombination nodules. So here the recombination nodules, it plays an important role in regulating the crossing over in between the non-sister chromatids of homologous chromosomes. Is it clear? In addition to the chiasmata, even we can observe one more other specific structure which is termed as recombination nodules. So here these recombination nodules, they play an important role in regulating the genetic exchange in between the non-sister chromatids of homologous chromosomes. Is it clear? Recombination nodules play an important role in regulating the genetic exchange in between the homologous chromosomes. So here, in between the two homologous chromosomes, in majority of the two homologous chromosomes, here we can observe only one specific chiasmata. But here, there are specific chromosomes at which we can observe more than one specific chiasmata. Is it clear? In case of the majority of the chromosomes, where the crossing over takes place, here we can observe only one specific chiasmata in case of one specific chromosomal pair. But here there are certain chromosomes at which we can observe more than one specific chiasmata. So let me write it here. In majority of the chromosomes, we can observe only one chiasmata. But there are certain chromosomes at which we can observe more than one chiasmata, right? So here this is regarding the packet-in stage, which is a part of the prophase 1 of the Meiosis-1, and then after we will be studying regarding diploid stage in detail. So let me arrange the things here. So after the packet-in stage, the next specific stage of the prophase 1 of the Meiosis-1, here it is nothing but diploid stage. So here during the diploid stage, the specific synaptonemal protein complex, which is responsible for regulating the pairing in between the two homologous chromosomes, it successfully disappears. Is it clear? The specific protein complex, which is termed as synaptonemal protein complex, it is responsible for regulating the pairing in between the two homologous chromosomes. So here the synaptonemal protein complex, it successfully forms during the zygote stage. But here when the cell division, when it successfully progresses into the diploid stage, the specific synaptonemal complex, it successfully disappears. Though the specific synaptonemal complex, it disappears during the diploid stage, but here the specific region where the crossing over has took place, even the specific region, it is observed during the diploid stage. Is it clear? So let me write it here. During the diploid stage, the disappearance of synaptonemal complex takes place. But the specific region where the crossing over takes place termed as chiasmata. It is observed even during the diplotin stage, right? The specific X-shaped structure termed as chiasmata, even it is observed during the diplotin stage. And here, in case of majority of the living organisms, the specific diplotin stage, it remains for 1 month. In case of certain living organisms like yeast, and even in case of the oocytes of the vertebrates, the specific diplotin stage, it lasts for 1 month. So let me write it here. In case of yeast and oocytes of vertebrates, the diplotin stage lasts for 1 month. But whereas in female humans, the diplotin stage lasts for 12 to 15 years. Is it clear? So here this is regarding the diplotin stage in detail, and then after we will be studying regarding diakinesis. So here what is diakinesis? So let me arrange the things here. So after the diplotin stage, the next specific stage of the prophase 1 or the Meiosis-1, here it is nothing but diakinesis. So here during the diakinesis, what exactly happens here is, during the diakinesis, the condensation of the chromatin material, it successfully reaches to the accurate levels. So initially the condensation of the chromatin material which begins during the leptotin stage. Now here the condensation of the chromatin material, it successfully reaches to the accurate levels during the diakinesis. And in addition to this, the specific X-shaped structure which has successfully formed in order to regulate the crossing over in between the non-sister chromatids of homologous chromosomes, which is termed as chiasmata. That specific chiasmata, it successfully disappears during the diakinesis. Is it clear? The specific X-shaped structure which is responsible for regulating the crossing over in between the non-sister chromatids of homologous chromosomes, here it is termed as chiasmata. So here the specific chiasmata, it successfully disappears during the diakinesis. And in addition to this, during the diakinesis stage, the disintegration of the nuclear envelope takes place. Is it clear? The disintegration of the nuclear envelope takes place, and in addition to this, even the various subcellular organelles like nucleus and nucleolus, they disappear. Is it clear? So let me write it on the board. During the diakinesis, the condensation of the chromatin material reaches to accurate levels. And here during the diakinesis, terminalization of chiasmata, terminalization of chiasmata takes place. Chiasmata takes place. And in addition to this, disintegration of nuclear envelope, nucleolus, nuclear envelope, nucleus and nucleolus disappear. Nucleolus and nucleus, they disappear, right? And in addition to this, during the diakinesis, the specific homologous chromosomes which has successfully participated in case of the crossing over. Now here these homologous chromosomes, they are ready to separate. Now here, in order to regulate the separation of homologous chromosomes, now here organization of meiotic spindle takes place. Is it clear? The specific homologous chromosomes initially will successfully participate in case of the crossing over. Now here the specific homologous chromosomes, they are ready to separate. So here, in order to regulate the separation of homologous chromosomes, here there is a requirement of organization of meiotic spindle. So here, by taking the advantage of the specific meiotic spindle, the homologous chromosomes, they are ready to separate. Is it clear? Homologous chromosomes are ready to separate by taking the advantage of the organization of meiotic spindle. Is it clear? So here this is regarding diakinesis, which is a part of the prophase 1 of the Meiosis-1. And then after we will be studying regarding metaphase 1. So here, after the successful commencement of diakinesis, with this, the prophase 1 of the Meiosis-1, it is successfully completed. So within no time, the metaphase 1 of the Meiosis-1 comes into the picture. Is it clear? So let me arrange the things here. So during the metaphase 1, successful organization of meiotic spindle takes place. So here, because of the successful organization of the meiotic spindle, the two homologous chromosomes initially will successfully participate in case of the crossing over. Now here, these two homologous chromosomes, they are capable to align at the equatorial region. Is it clear? The two homologous chromosomes initially will successfully participate in case of the crossing over. These two homologous chromosomes, by taking the advantage of meiotic spindle, they are capable to align at the equatorial region. Is it clear? So let me write it here. During the metaphase 1, organization of meiotic spindle takes place. Right? Takes place. And here, by taking the advantage of meiotic spindle, the two homologous chromosomes are capable to align at the equatorial region. Right? So here, this is regarding metaphase 1. After the successful commencement of metaphase 1, the next specific stage period is nothing but anaphase 1. Is it clear? So let me arrange the things here. And during the anaphase 1 of the Meiosis-1, the two homologous chromosomes initially which has aligned at the equatorial region, these two homologous chromosomes, they are ready to migrate towards the spindle pole during the anaphase 1. And here we need to understand that in case of the anaphase 1, segregation of homologous chromosomes takes place, but not the separation of sister chromatids. In case of the anaphase of the mitotic cell division, separation of the sister chromatid takes place, because in case of the anaphase of the mitotic cell division, splitting of the centromere takes place. But whereas in case of the anaphase 1 of the meiosis 1, here the splitting of the centromere does not take place. So the reason why instead of the separation of the sister chromatids, here the segregation of homologous chromosomes takes place. Is it clear? So let me write it here. During the anaphase 1, segregation of homologous chromosomes takes place. Homologous chromosomes takes place. The specific homologous chromosomes initially which are at the equatorial region, they start migrating towards the spindle poles. Right? So here this is regarding anaphase 1 of the meiosis 1. So here in case of the anaphase of the mitotic cell division, in general, separation of the sister chromatid takes place. So let me show it here. In case of the anaphase of the mitotic cell division, in general, separation of sister chromatid takes place, because in case of the anaphase of the mitotic cell division, in general, splitting of centromere takes place. Because of the splitting of the centromere, separation of sister chromatids, it is observed. But whereas in case of the anaphase 1 of the meiosis 1, here the splitting of centromere does not take place. So the reason why, instead of regulating the separation of sister chromatids, here the segregation of homologous chromosomes takes place. Is it clear? So here this is regarding anaphase 1 of the meiosis 1 in detail. And then after, the next specific stage period is nothing but telophase 1. Is it clear? So let me arrange the things here. And during the telophase 1 of the meiosis 1, after the successful segregation of homologous chromosomes towards the spindle pole, now here the specific nuclear envelope initially which undergoes disappearance during the diakinesis stage, again the specific nuclear envelope, it successfully reappears during the telophase 1. And here we need to understand that during the telophase 1, here we cannot observe the complete decondensation of the chromatin material. So the reason why, again the specific chromosome, now again it has to participate in meiosis 2. Is it clear? During the telophase 1 of the meiosis 1, though the specific chromosomes, they successfully reach to the spindle pole, but here we cannot observe the complete decondensation of the chromatin material. So the reason why, here these specific chromosomes, again they has to participate in one more other specific cell division, which is nothing but meiosis 2. Is it clear? So let me write it here. During the telophase 1, after the segregation of homologous chromosomes, now the nuclear envelope reappears during the telophase 1. And here, in order to acquire in order to acquire the decondensation of the chromatin material, the specific chromosomes has to enter into prophase 2 of meiosis 2. Prophase 2 of meiosis 2. Is it clear? So here, this is regarding the Meiosis-1, which is a part of meiosis and its regulation in detail. And then after, in the next upcoming lecture session, we will be studying regarding meiosis 2 in detail. So here, I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends.